Okay, chapter 5, vision. The next three chapters are really dealing with sensation and, not perception, but sensation, sensory systems, and movement. So for this chapter, it's going to be short. There's 3 modules to the chapter. We're only going to deal with the first module. Really the basic structure and function of vision and how it operates and how the eye detects energy. Laying it out for how all our senses, smell, taste, hearing, respond to the outside world. There's physical stimuli that exist out there in the world, and our bodies must be able to recognize that. So let me show you this real quick. So we say physical stimulus, we're speaking about, for vision, it would be light waves. For sound, it would be sound waves. Touch or feeling, prick on the skin, taste, smell, molecules in the air. Those are physical stimuli. So the body must recognize that. So physical stimulus is really external, outside of our body. When it comes into our body, the body must now process it. So the physiological response is going in the body to process light, hearing, taste, sound. Neuroactivity is occurring. That is, something is actually going on in the cell to process that stimuli. Once it's processed, now you have a sensory experience. So put under this physical stimulus, response, and now you have the experience. So the experience really is a subjective feeling. Some people like certain colors, some people like certain sounds, certain tastes. So now it becomes subjective. The point is that from the physical stimulus, which is external, the body now must process that. So for vision, the physical stimulus for vision is light or photons of energy. I'm going to show you this first in the PowerPoint slides that rolls through the discussion on vision and topics. I'll show you some other things beyond just the PowerPoint, but essentially vision and visible light. If you see this large spectrum right here, these colors, this whole range here is the electromagnetic spectrum. Where you see those colors, those are the visible lights that we see. Those are the photons of energy that we detect. The measurement in that span is about 350 nanometers to about 750 nanometers. Nano, N-A-N-O. Nano means billionth meter, billionth of a meter. Really small. So 350 on this end, you got like purple leading to blue, green in the middle, and on the other end, the longer band or the longer wavelength is 700. It's around red, bright red. So that's visible light, a small portion when you blow it up, but there's a large spectrum out there. You got on one end AC circuits in terms of plugging in appliances in to get electricity. Broadcast bands, getting your radio signals. You can listen to a program on your radio in your car. You got radar. Radar is on the end of using some technology that can detect things in terms of speed. So it's a certain kind of light wave that can be detected. Radar and infrared rays. These are then close to what we're talking about. You ever seen that movie Predator? And it's seeing in like heat sensing. So there's certain kinds of auras that are around people, and that heat is what some people can visualize with a visible light. And on the other end, other extreme, which is kind of dangerous, ultraviolet rays coming from the sun can destroy the skin cells. So ultraviolet radiation can be problematic. You get the X-rays, it's problematic for getting too many X-rays because it can destroy your DNA in your body. Gamma rays, again, something out there in the cosmic elements generating these pulses of energy that could have an impact on the body. So On the extreme of this end, dangerous. On this end, not so much dangerous to the body, but our bodies respond to all that. But think of spiritual waves and conscious waves. Are they in that element too, where the body's detecting that? Potentially. But for us, in the topic on vision, we're only speaking about one little span of space. So the eye, how is the eye constructed? There's plenty of material you'll see in your book, but let me go ahead and explain this particular image right here. Here is a physical stimulus or an object, a leaf. The leaf must be somehow put onto the eye to detect that that's what it is, a leaf. Not even dealing with the brain yet, we're just dealing with the eyeball. So the leaf must somehow get on a part of the eye that's detecting what it is. So let's look at structure first. On the outside of the eye, you have a cornea. In the middle part of the eye is a pupil. Right around the pupil is the iris. Iris opens and closes. That means it makes the pupil bigger and smaller. Beyond the iris is this thing called the lens. The lens can be oblong or concave, convex or concave. It can change its shape to help the image get onto the part of the eye that's detecting vision. So there's fluid in here and there's fluid in here. So the waves have to get through the fluid. It's called refraction. So on the back part of the eye is the retina. Back part of the eye is the retina. On the retina, there's a very important place where this object must get to the retina. That place is called the fovea. So that object right there must somehow get onto the retina at that place called the fovea, the central point. So the fovea is a place where it has the highest visual acuity, that is sharpness. So the image must get onto the fovea. So on the back of the eye, the retina then makes up the optic nerve that then goes to the brain and detect, oh, that's a leaf. Now you're able to process what it is. You might have thought it was a gun. There are many people thinking that things are guns and shoot people. Seeing the wrong things. The brain processes that. Eyes is detecting the actual image that the brain processes what it's seeing. So if you look at this, because we're going to get through this quick, you can look at the PowerPoints on your own. Eyeball, that's the cornea on the outside. Guess what? It could get scratched. Some people have corneal transplants. You could have a cracked windshield. And a cracked windshield is like a scratch on your cornea. It's always there. Some people can have it repaired and it can go away in terms of the scratch, but that's the outside covering cornea. Take this apart. So all these blood vessels right here are feeding the eyeball. This right here, these are muscles. Because your eyes can what? Go up, down, side, can move around, so the muscles do that. There's the pupil, right in the middle. Take this out here. Pupil's in the middle, and this gray area here is the iris. So as it opens and closes, it makes the pupil bigger or smaller. So when you go to the doctor and they're putting a light in your eye, the light is shining right there, and of course the pupil's going to get smaller. If it's dark outside, the pupil gets bigger because it wants to make sure it's seeing things, because that's to get back to what? That retina. So iris, pupil, pick this apart, and look inside. There's the lens right behind the iris. Take the lens out, look at the shape of the lens. So depending upon the shape, helps to refract the image so we can get to the back part of the eye where the retina is. So again, here's the lens, take it out. Inside here is fluid. You see on the back side of there, blood vessels. And then on the back of the eyeball is where the retina is located. We'll see some images to show that. And then the retina has cells that then combine, go to this optic nerve, which then goes to the brain. So that's the structure of the eye. So it does say on there blind spot. So there's a little indentation where the fovea is, but where the indentation is, there's a lot of cells. But right here, the blind spot is where there's no cells. And that's why it's more difficult for someone to see at that location because there's no cells there. So you really could have a blind spot in your visual appearance. So if you think about the eyeball and why some people wear glasses, there's a lens. Depending upon the shape, depends upon if the image is going to get onto the retina. So right here is the light rays from a distant object. Right here, it cannot focus clearly because the dotted line shows that it's not reaching the back part of the eye, the retina. So to correct that, you need glasses, or the eye will be not seeing right. So the corrective lenses are like your lens in your eyeball, but they're correcting the way that the light is shining onto the eye. So now with the corrected lens, it shines right onto the fovea, right onto the back of the retina. Here, it's right here, it's overshooting the retina. It's going on the back side of the retina. But to have clear focus, the lens could change, and now it gets directly onto the retina. So again, the lens and its shape depends upon how that image is going to get onto your retina. And again, that's why people squint or they wear glasses, because they're correcting the way the light is getting onto the retina. We'll come back to this. So when you look at your PowerPoint slides, essentially just the first module, 5.1, it's just saying that there's a term called law of specific nerve energies. Law of specific nerve energies. Every particular nerve pathway responds to a certain kinds of stimuli. That stimuli could be vision, therefore the eyes are detecting vision, not smell. For the ears, they're detecting hearing, not touch. So the law of specific nerve energies is making reference to a specific nerve for a certain kind of receiving of energy. So you don't have it mixed up. At the very end of the chapter, however, there is a topic called synesthesia. Synesthesia, where your actual energies or your perceptions are mixed up. As if you could like smell sounds or hear colors. Yeah, that can really happen for some people. So the term is called synesthesia. You can read up on that at the end of the chapter, but we're not going to emphasize it too much. Because the reality is it an abnormality or is it an enhancement in the person's experience? So again, if you look at the PowerPoint, it tells you about light. It just broke it down with the images and the model, but it tells you how light gets from the outside to the eyeball. So you can read up on that. And the back of the eyeball becomes important. Another figure, just like I just showed you, you can look at that again. You see all these, has muscles right here. You have muscles that may be even controlling that lens. So if they come out long or concave, depending upon how it is going to depend upon how the image gets onto the back of the eye. You saw in the beginning of the chapter, hopefully you read that, but there's a bird looking like that. The question is, why would the bird have to look like that? So that's a predatory bird, like a hawk, like a falcon, like an eagle. Where do they hang out? Up above. Telephone poles, trees, flying high, looking down. So if they're looking down, for them, the place for highest visual acuity, their foveas are pretty much on the top part of their eyeball, the top part of their retina. Not like for us straight ahead, but for them it's further up. So as they're flying above, the image is hitting on to the top part of the eyeball as they're flying above. But if they come down to our level and are sitting right in front of us, they really can't see. So to turn the head to try to get that image onto that place that's highest visual acuity for them. You know, birds can fly high, and also predator birds can really see at a distance because they actually have two foveas that exist to give them highest visual acuity. So on the back of the eye, understand this. Light shines all the way through the retina, hits the back of the eye, then information is processed to go to the optic nerve. So light shines all the way through, hits the back of the eye, then the cells are processed this way before it then goes to the last group of cells to go to the optic nerve. So the cells that are in the retina will be the amacrine cells, bipolar cells, horizontal cells, ganglion cells. I would like you to memorize, or really recall the ganglion cells, because they're the last group. Ganglion, the last group that leads to making the optic nerve. So again, horizontal, amacrine, bipolar, ganglion, all those cells are in that retina, processing information, so you can now interpret what you're seeing. The very back of the eyeball is pigmented. Pigmented, like, yes, with color. Without that color, you're virtually blind. People have a disease, some people have a disease called retinitis pigmentosus, where they have absence of pigment on the back of the eyeball. Everything they see is blinding. It's understandable. White reflects, black absorbs. So as the pigment on the back of the eye is absorbing, it's taking in the energy, processing. If it's not black and dark, it reflects and you're basically blind. So it's important to have that pigment on the back part of the eyeball. So the optic nerve again is very important. It's actually one of the largest cranial nerves. Remember, you have 12. The optic nerve number 2 is the largest, one of the biggest. Talk about the blind spot. You can do example on your own where there's like a little X that you see. You stare at a certain point, and if you keep going in and out, that X will disappear because it'll be getting onto that part of the eyeball where there's no receptors, just the blood vessels. So that's the blind spot right there. That's why sometimes when you're seeing things, they're not in your vision. So again, fovea, recall what that is. You'll be asked that question later. So here we have these predatory birds. These are like owls. You can see They're trying to look to see where they are. If you think about pigeons, they're not predatory birds, but they can walk around on the street and you can walk past them and they can see you. They're not worried about this. So they don't have to have their phobias up top. Their phobias are processed where they can operate in that kind of environment. But predatory birds, their phobias are so they can attack from above. So for the retina, you think of central and peripheral. You have cells in the central part of the eyeball. When we say central, we're meaning like around the middle part. So if we say central, we mean like around the middle. If we say peripheral, we mean further outside. So this is the eyeball, half of it. Central is here on the inside middle. Peripheral is further out, away from the central part. So the cells that are in the central part are pretty much cones. Cells in the peripheral part are pretty much rods. So let's deal with this because the retina contains the actual receptors that are actually detecting the image. And so this table right here gives you a sense of how to compare rods and cones. So cone, central, rods, peripheral. However, you have almost 120 rods to about 6 million cones. You have more rods than cones. So more rods in that periphery, cones centralized, but they're on top of one another and help out with clear, detailed vision. Dim light rods outside, night vision. So about 120 million rods to about 6 million cones. Rods, dim light, night vision. Cones, daylight, clear vision. Shapes. The rod has a kind of like flat end cone, like an ice cream cone. That's why I think the names rods and cones. On the very end, there's some disc, because that's why it's kind of striated. Disc, like seed disc. In those discs are photopigments. Photopigments. Chemicals in those discs that help to process light. So chemicals says 11 cis retinol bound to proteins called opsins. Light energy converts retinol quickly to all transretinal. Sounds kind of confusing there, but let me see if I can do this for you. Because some of the chemicals have some technical names, but rhodopsin is one particular photopigment. In the presence of light, rhodopsin can break down into, I believe, opsin, and retinol. Presence of dark, it then goes back to rhodopsin. So light and dark change these chemicals to different processes. So the retinol, like that 11 cis retinol that was in that other slide, is a particular chemical that's related to it that we can eat, consume. things like carrots, beta carotene. Carrots, beta carotene pigments. So those pigments are supposed to be very helpful for vision. So color vision. So there's some theories on color vision. We talked about this already with the spectrum, large magnetic spectrum. So one of the theories is that they have certain kinds of cones, because they're color vision. Cones detect certain types of wavelengths. The red, long, blue, short, green, yellow in the middle. Different cones for different wavelengths. So when you look at this, it's called the trichromatic theory. Trichromatic theory. The two people who related their work to it, Young and Helmholtz. Over the time, they weren't in the same time period, but Young and Helmholtz and their research have thought about the different types of cones, red, green, and blue, and then those cones are what are helping us to process color vision theory. Another one is the opponent process theory. Opponent, opposites process. Almost like if you see black and white put together would create gray. Opponent process. That's what that one's talking about. Again, theory. This is happening in the retina. So keep that in mind. You can read up on V2 because I'd like for you to just recall what the basics are for them. So trichromatic, 3 different wavelengths of light, 3 different cones detecting that. Image showing you how that can be done. You can read up on that on your own. Opponent process, again, showing that the opposite wavelengths, the paired colors give you something different. Like an after image that could occur, and that's how we were thinking about. If you look at something bright, close your eyes, you still see the image, which speaking about that, how the opponent process is also creating that color. And then the last one here is the Retinex theory. Retinex theory. So the 3 theories: trichromatic, opponent process, and Retinex theory. So now this goes beyond just the eyeball detecting color. It's now the brain, cortex. That's why it's combining retina and cortex, Retinex. So if you see, let's say, a White House, like the White House in DC, and there's green film over the White House, is the house green or is it white? We know it's a White House, so it should be white. It's just a green film over it. So your brain is now processing that. So Rubik's Cube, just an example of, you know what it looks like. You put a film over it, blue, well, you know what those colors should be, but your brain is processing something different. Again, theories. So the last part of the chapter pretty much gets into perception. We're not going to go over perception. This whole class is on perception. So what we'll do here is just focus on the issue of how the eye is actually detecting images. Certainly there's some problems with people's vision, be nearsighted, farsighted. You have astigmatism, nearsighted, problem seeing distance, farsighted, problem seeing close. So you've got people that need glasses for reading, some people need glasses for distance. So the corrective lenses deal with that. Some people have multiple sclerosis. There could be a problem with the optic nerve, which is highly myelinated. So here's the nerve coming off, all that white stuff around it is the myelin. Inside is the actual tissue in the nerves. If that myelin is deteriorating, a person has a problem with focal vision, can't see clearly in front of them. People can be born blind, and that could be problematic. But the reality is that you got to take care of your eyes. When you go to the doctor and the ophthalmologist, and they're testing your eyes, put a puff in your eye, see how your eye responds. Sometimes that's needed to see if you have glaucoma. What is glaucoma? Glaucoma is pressure on your retina. Not a good thing. If the pressure's on the retina, it's squeezing the cells and tissues, and you can potentially become blind because of that tension. That's why hypertension, diabetes, all that can be related to problems later on, and your eyes can be also a problem, have a problem. So again, take care of your eyes. In terms of our topic of discussion, essentially, we just want to make sure you understand how the eye operates in terms of processing vision. For the brain, just remember that the occipital lobe is where the vision actually is being processed. Not in the front of the head, but back of the head. Okay, so next chapter we'll focus on other sensory systems.